Hey everyone, Dr. Steve here. You know it as well as I do, but most students need a B or better in A&P. But did you know that students are doing that all over the US using my Anatomy and Physiology Digital Suite as a low-cost alternative to expensive textbooks? Digital Suite delivers the key concepts of A&P with short, assignable tutor videos and a bulleted narrative study guide that not only explain the concepts of A&P, they show them happening. Combined with state-of-the-art dissection and physiology lab simulations and a vast question bank for quizzes, homework assignments, and exams, Digital Suite meets students where they are and gives them the tools to master the content. It's the perfect supplement to an OER textbook or just on its own. If you're an A&P instructor, contact your McGraw-Hill representative today and ask about Anatomy and Physiology Digital Suite. If you're a student, tell your instructor about it. There's a link to Digital Suite in the show notes of this episode. Get your B or better in A&P with A&P Digital Suite. Hey everyone, welcome to episode 3 of my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan, coming to you from Bucks County, Pennsylvania, just outside of Philadelphia, where I've been teaching A&P since 2002. Today begins our discussion of cells. We'll focus this episode on the plasma membrane of the cell, which is the boundary between the cell's interior and everything outside the cell. I know that you're probably thinking, hey, this is anatomy and physiology. I'm here for muscles and bones and organs and all that cool stuff. But most anatomy and physiology classes start with cells, because in order to understand all the organs and tissues, you really have to understand the smallest parts that they're made of. And those things are cells. Specifically, since humans are animals, we're going to talk about animal cells. We'll get to that in a second. I just wanted to thank you for following the show and listening. I know that there was a bit of a learning curve getting the podcast up and running on all the podcast platforms, but I think I've got it figured out now, so you should see the feed coming right up. I intend to make regular episodes to keep you up on the content so you can succeed in your A&P course, or you can simply be informed about the human body if that's why you're here. I'm actually traveling again. I'm recording this from a hotel in Chicago. Tomorrow, I'll be doing 2 separate presentations with local colleges here in Chicago about how to utilize digital technology in their anatomy and physiology classroom. So that's going to be a lot of fun. I will say this about the hotel I'm staying in. It is a little strange. The music in the lobby is so loud, and I'm sitting in a room with no carpets, kind of these weird tile walls, no desk, only a few hangers to hang clothes on, no drawers to put clothes in, and this really kind of weird vibe. I mean, I don't want to mention the hotel's name, but it is definitely different. So anyway, let's get moving on with cells. The study of cells is called cytology, Cytology. That's not Tom Cruise's religion. That's Scientology. Sounds similar, not the same. This is cytology, the one that is actual science, doesn't just have science in the name. So keep those things separate for sure. Cytology is the study of cells. You'll often see cyto, C-Y-T-O, as a prefix. You'll also see cyte, C-Y-T-E, as a suffix for things. For instance, erythrocyte, red blood cell. Monocyte, a type of white blood cell. So those are where you'll kind of see those things come in. Every living thing that we know of is made of cells and the materials that those cells produce. Cells are the smallest units of life that are considered to be alive, and they come in lots of shapes and sizes. You can see flat, tile-shaped cells or round cells, square cells, rectangular cells, some cells that look like stars. They all look different. Some cells are really long, like a neuron could be a meter long. And then some cells are really, really tiny. Some cells have a nucleus and some don't. Some have more than one. So they're very different from each other, but they all come from the same single cell in your body that is derived from the one sperm cell that fertilized the one egg cell that eventually became you. Your embryonic development started with that fertilization. A cell with half the number of chromosomes in it fertilized another cell with half the number of chromosomes in it, and the combination of those chromosomes in that nucleus eventually became a full cell that divided and divided and divided, leading to all of the cells that exist in your body today. Well, not all the cells. Actually, there are 50 trillion bacteria that live in your body that are not you, but they're also there. They just live inside your digestive tract. So it's kind of interesting the way that happens. Now that you're born and fully developed, you've got tens of trillions of cells that all come together and, with cells that are like them, form tissues. Those cells all develop and differentiate from that one single set of stem cells. Those original cells had no specialty. They had no special function. They had no special look to them. They're what we call undifferentiated cells. But eventually, those cells will start to develop and differentiate into skin cells and bone cells and liver cells and kidney cells and so on and so on and so on. That's when they have specific functions that contribute to your overall body function. Those like cells will come together to form tissues, and then those tissues will come together to form organs, and the organs come together to form organ systems, and that's what does the jobs of your body. And all those organ systems become the organism that is you. But it all starts with cells. There are cells that secrete hormones. We call those endocrine cells. We've got cells that start off long and then get shorter so we can move our joints. We call those skeletal muscle cells. We have cells that can conduct electrical signals like neurons and muscle cells. The cells of our nervous system utilize this electrical activity so that we can experience the universe. Okay, so what makes animal cells different from other cells is that the outer boundary of an animal cell is not a wall, it's a membrane. There are two main parts to the cell. There's the outer boundary, which we call a plasma membrane in an animal cell, and there's the inside called the cytoplasm. The cytoplasm is a mixture of a lot of different things. There is a fluid called cytosol, functional components called organelles, and non-functional components called inclusions. Organelles do the individual jobs that all come together so that the cell can do its big picture jobs. You've heard of organelles. They are mitochondria, Golgi complex, endoplasmic reticulum, and others. I'm sure you've heard of those. We're going to talk about them later on in another episode, but I want to focus on the plasma membrane for now. It's really what makes us different from plants and bacteria and fungi. The plasma membrane is made mostly of lipids, whereas the cell wall of plants consists of a complex carbohydrate called cellulose. In addition to the lipids, our plasma membrane has proteins. I want to focus on the plasma membrane, but in order to do that, we have to talk a little bit about chemistry because the chemical properties of the lipids in the plasma membrane and the water in the cytosol are incredibly important. Some molecules, like water and glucose, are what we call polar. That means that the charges of the atoms in that molecule are not equally dispersed throughout the molecule. Even though the overall charge is zero, the protons and electrons are not evenly dispersed across the molecule. So part of the molecule is a little bit positive, and the other part is a little bit negative. So essentially, the molecule has poles, like a battery. Polar molecules are what we call hydrophilic, which means they have a tendency to mix with, dissolve in, or be wetted by water. Some people use the term water-loving. That may help you remember what hydrophilic means. Sometimes you'll see the term water-soluble. I use that a lot, which also means hydrophilic. So polar molecules are water-soluble. But then we also have nonpolar molecules whose charges are evenly dispersed across the molecule. We call them hydrophobic, which means they have a tendency to repel water but dissolve in lipids. Lipids are fats like oils. Polar molecules, on the other hand, do not dissolve in lipids. So if you try to mix a polar molecule with a fat, you know what happens. An example of a polar molecule is water. Think about oil and water. Oil does not dissolve in water and vice versa. They remain separated. These are different classes of molecules. Like I said before, I tend to use the term water-soluble and lipid-soluble because it kind of helps with the context of where we are. So why is this important to us? Well, because most of the plasma membrane is made of lipids, about 98% of the molecules in our plasma membrane are lipids. 75% of those are called phospholipids, and that means they're lipids with a phosphate group in their molecule. They look kind of like a big round head with two skinny tails. The head is the hydrophilic phosphate group, and the tails are made of hydrophobic fatty acids. Since this molecule, these phospholipids, have a hydrophilic end and a hydrophobic end, we call them amphiphilic. It means that they've got a water-soluble part and a lipid-soluble part. Each plasma membrane of our cells has two layers of these phospholipids, so we call it the phospholipid bilayer. And they're arranged based on their affinity for water, which leads us to the second reason we're concerned with solubility. The extracellular fluid outside the cell and the cytosol that is inside the cell are both mainly water. Okay, so consider the bilayer. Two layers of phospholipids, one contacting the fluid outside the cell, and the other contacting the fluid inside the cell. The hydrophilic heads of the phospholipids will be attracted to water, so phosphate heads of the outer layer face out, and the phosphate heads of the inner layer face in. This leaves the hydrophobic fatty acid tails in between the phosphate heads. And this is our phospholipid bilayer. Think of the phospholipid bilayer as two hairbrushes facing each other, so the bristles of one are touching the bristles of the other. The bristles are the fatty acid tails, and the back of the brush is the phosphate heads on each side. It's not perfect because the phosphate heads do not make a solid backbone. They remain separated from each other, but hopefully this helps you get a mental picture of what we're talking about. Now keep in mind that the plasma membrane is not a rigid barrier. It's fluid and being held together mostly by the hydrophilic and hydrophobic forces. The phospholipids move around and spin, changing their location in the membrane. The attraction to water keeps the hydrophilic heads facing the extracellular fluid on one side and the intracellular fluid, cytosol, on the other side. The repulsion from water keeps the fatty acid tails in the core of the membrane, avoiding the water on both sides. Okay, only about 75% of the lipids in the membrane are phospholipids. That means that there are going to be some others. 20% are cholesterol molecules. Not the same kind of cholesterol that's in the blood. Those are lipoproteins with cholesterol in them. A cholesterol molecule is a vital, waxy lipid with a unique structure featuring a 4-ring steroid nucleus. Now, in addition to that steroid nucleus, there is a hydrocarbon tail and a small hydroxyl group. This structure makes it both hydrophobic and hydrophilic at the hydroxyl end, allowing it to be embedded in the plasma membrane to provide structural support by stiffening the membrane, regulating fluidity, and acting as a crucial precursor for building steroid hormones like testosterone and estrogen and vitamin D. The cholesterol you hear about clinically is actually lipids and proteins bound together and is used to transport cholesterol in the blood. We'll discuss that in a later episode. The remaining lipids are glycolipids. These are phospholipids with carbohydrate chains that extend from the extracellular surface of the membrane, forming the glycocalyx, which is a fuzzy coat that functions in cellular identity. These carbohydrate identity markers are how your immune cells recognize your own cells so they don't attack them. Since we're on the topic of the lipid bilayer, let's talk about how that applies to the movement of materials into and out of the cell. So the reason why we're so concerned with the hydrophilic and hydrophobic nature of molecules is because the lipid bilayer, or the plasma membrane itself, one of its jobs, in addition to just being the boundary that holds the cytoplasm in, is that it has to regulate the movement of materials into and out of the cell. And that's a really important job. Our plasma membranes are described as selectively permeable. The word permeable means allowing liquids or gases to pass through it. Because our lipid bilayer is hydrophobic, some things can get through and some things can't. So that's kind of a problem for us. we have to consider the type of molecules we need to get in and out of our cells. Are they lipid-soluble or are they water-soluble? If they're lipid-soluble or hydrophobic, they can diffuse right through the plasma membrane's lipid bilayer. If they're hydrophilic or water-soluble, they can't. The lipid bilayer is a barrier against hydrophilic substances. So these hydrophilic water-soluble substances just bounce off the barrier unless the molecule is really small, like water or urea. Water only has 3 atoms in a molecule of it. Small molecules like that can sneak through the gaps between the fatty acid tails, but large polar molecules like glucose can't get through the lipid bilayer on their own. They need some help, and we'll discuss how that happens. Okay, so that's why it's really important to understand the chemistry of the plasma membrane, hydrophilic and hydrophobic materials, the lipid bilayer, and all that goes with it. But there's more to the plasma membrane than just lipids. Dispersed throughout the bilayer are several kinds of protein-based structures. We call them membrane proteins. And the membrane proteins can either be embedded in the phospholipids, we call those integral proteins, or they could just be floating along the inner or outer surface of the membrane. We call those peripheral proteins. Membrane proteins have very important functions. Some allow the cell to respond to chemical messengers in the area, some are used for adjacent cells to attach to one another, and some allow non-permeable substances to pass through the membrane. Ions like sodium ions, potassium ions, chloride ions, they can't permeate the membrane lipids, so they need help to make their way into and out of the cell. There are several different kinds of membrane proteins, and we'll discuss them all, so that way you have a good understanding of their function. Now, some of these integral proteins, the ones that are embedded in the phospholipids, go all the way through so that they have a surface facing the outside of the cell and a surface facing the inside of the cell. We call those transmembrane proteins because they go all the way through the membrane. Okay, let's look at some examples of membrane proteins. First, we'll talk about receptors. Receptors allow the cell to respond to a chemical messenger that's outside the cell or to a chemical messenger that's inside the cell. For example, if you have a receptor for a particular chemical like acetylcholine, which is a neurotransmitter, you'll have a membrane protein in the cell's plasma membrane that will chemically react with that acetylcholine and then cause something to happen to the membrane or inside the cell. Like maybe an ion channel will open, for example. And if an ion channel opens, that means that ions can use that channel to flow into or out of the cell. We use receptors so that the cells can respond to hormones, neurotransmitters, mechanical stimuli, etc. There's a lot of different things that receptors can respond to, and each receptor is specific, meaning that a receptor will only respond to that one stimulus. For example, a receptor for insulin will not respond to acetylcholine. This is because they're chemically specific and they chemically react in a very particular way so that only that chemical can stimulate it. We call those chemicals ligands, or sometimes you'll hear people say ligand.
Nobody does selling better than Shopify. Shopify is the home of the #1 checkout on the planet. And the not-so-secret secret, with Shop Pay that boosts conversions up to 50%, meaning way less carts go abandoned and way more sales going through. So if you're into growing your business, your commerce platform better be ready to sell wherever your customers are scrolling or strolling. On the web, in your store, in their feed, and everywhere in between. Businesses that sell more, sell on Shopify. Upgrade your business and get the same checkout that Skims uses. Sign up for your $1 per month trial period for 3 months at shopify.com/Westwood1, all lowercase. Go to shopify.com/Westwood1 to upgrade your selling today. Shopify.com/Westwood1.
Another type of membrane protein is enzymes. Enzymes can catalyze an array of chemical reactions, including digestion and hormone action. So there's enzymes that catalyze chemical reactions embedded right there in the plasma membrane. There's lots of things those enzymes could be doing. Sometimes they're involved in what's called a second messenger system that allows non-permeable chemical messengers to create effects inside the cell. Lactase, the enzyme that breaks down milk sugar, is another example. As this podcast goes on, we will cover many enzymes that take care of an array of chemical reactions inside the body. So you heard me mention just now ion channels. Ion channels are a specific kind of membrane protein, and what they do is they act like tunnels that go right through the phospholipid bilayer and allow substances that wouldn't normally be able to get through the phospholipids to get through. They often allow water-soluble molecules to pass into and out of the cell. Sometimes those channel proteins are ion channels and they allow ions to pass through. Sometimes they're aquaporins and they let water pass through in large amounts. Now we know from before that water is such a small molecule that it can actually slip through the gaps in the phospholipids in small amounts. But in larger amounts, they go through channel proteins called aquaporins. Think of the word aqua for water and porin for porous. We also have membrane proteins called carriers that will bind to water-soluble molecules and carry them into and out of the cell. We have cell identity markers. These are membrane proteins that have carbohydrates attached to them, and that carbohydrate contributes to a fuzzy coat on the surface of the cell called the glycocalyx, which I mentioned earlier. Proteins with carbohydrate chains attached to them are called glycoproteins. The reason they're called cell identity markers is because they have specific antigens on them, which are little chemical markers on the cell that are specific to each individual person or organism. Its purpose is to allow the body's immune system to recognize that cell as part of its own body. That's a really important thing to do because you don't want your immune system attacking your own cells. Your immune system is sending cells that go throughout your body that are there to monitor and see if there's any kind of foreign cell or virus or bacterium in your body that it needs to disarm so it doesn't make you sick. So what we want to make sure is that your immune system isn't attacking your own healthy cells thinking they're foreign cells. So your cells have this structure called the glycocalyx, which is made of carbohydrates sticking off of these membrane proteins and phospholipids. If the immune system doesn't adequately recognize your own cells, it will attack your own healthy cells, and that is called an autoimmune disorder, like lupus or rheumatoid arthritis, and those are bad, so we definitely don't want that happening. Another kind of plasma membrane protein is cell adhesion molecules, or CAMs. They form what's called cell junctions, which is how cells bind to other cells, forming tissues. Cell junctions also bind cells to other extracellular materials like the basement membrane. So using these proteins and using the hydrophobic nature of the lipid bilayer, the plasma membrane not only acts, like I said before, to form a boundary for the cell and keep the cytoplasm on the inside, it also regulates the flow of materials in and out. And that's a good thing, because we don't want too many sodium ions coming into the cell at the wrong time, or potassium ions leaving the cell at the wrong time, or vice versa. It all has to be very well and tightly regulated to maintain optimal health. Okay, lastly, let's talk about structures that extend from the plasma membrane, namely microvilli, cilia, flagella, and pseudopods. Microvilli is the plural of microvillus. They increase the surface area of the plasma membrane. We see the best examples of microvilli in the small intestine because they maximize a cell's capacity for absorption. Let's take a small sidestep to explain the surfaces of cells that line the spaces of our bodies. Every surface of your body adjacent to a space is lined with a type of tissue called epithelial tissue, which is made up of epithelial cells. It could be an internal surface like your digestive tract, your respiratory tract, your urinary tract, your reproductive tract, or an external surface like your skin. Epithelial cells have their own parts or surfaces. The apical surface is directly adjacent to the space that the cells are lining. The basal surface is opposite the apical surface and attaches the cell to an underlying tissue called the basement membrane. The sides are called the lateral surfaces. Cells with microvilli line the lumen of the small intestine. A lumen, by the way, is the space inside a hollow organ or vessel, like the small intestine. Microvilli are only about 1 to 2 micrometers long. So in your A&P lab, you probably don't have microscopes with high enough resolution to see them individually. Sometimes they'll show up as a darkly stained band across the apical surface of a cell. The microvilli make the apical surface of the small intestine like the surface of a hairbrush. In fact, it's called the brush border. Microvilli hold their shape by way of protein filaments called actin found inside them. That actin anchors to other protein filaments inside the cell called the terminal web. Some microvilli have sensory functions. We see them in the taste buds and the ears. We'll get into their detail much later in the podcast. Next, we have cilia. Cilia are hair-like processes that are up to 10 micrometers long. At least one cilium is found on almost every cell in the human body. It often serves a sensory function and doesn't move. So we call it a non-motile primary cilium. Among other things, the primary cilium contributes to smell, balance, vision, fluid balance, and gated ion channels. The cilia that move are called motile cilia. We don't see them in as many parts of the body, but when we do, they are in large numbers on each cell's apical surface. We find motile cilia in the respiratory tract, uterine tubes, which were formerly known as fallopian tubes, the ventricles of the brain, which are the cavities where cerebrospinal fluid is formed and flows, and in the testes. When these motile cilia move, they create wave-like motions in the same direction across the apical surface of the cells. This helps propel the extracellular contents in whatever lumen or cavity they're in. For example, after an egg cell is ovulated by an ovary, it travels through the uterine tube toward the uterus because the beating cilia create waves of mucus in that direction. One beat of a cilium is called a power stroke. Adjacent cilia beat consecutively like falling dominoes to create a wave-like motion. Now, if there was only mucus in the lumen, the power strokes of the cilia wouldn't be strong enough to move it. They'd get stuck in the sticky mucus. So membrane proteins called chloride pumps pump negatively charged chloride ions into the extracellular fluid of the lumen, right on the cell's surface. Positively charged sodium ions are attracted to chloride, so they follow. The concentrations of these ions lead to the addition of water via osmosis. The sodium chloride and water create salt water, also known as saline, and that saline sits directly on top of the cell's apical surfaces. The mucus floats on the saline while the tips of the cilia move it along. You may have heard of cystic fibrosis, a genetic disease that disrupts the addition of chloride pumps to the plasma membrane. Without the saline layer, it is too difficult for cilia to function in the respiratory system, disrupting the ability to keep the airway clear of excessive mucus and inhaled foreign particles. And that's not the only thing. You also see cystic fibrosis affecting the pancreas and its delivery of digestive enzymes to the small intestine and other areas. But it's pretty bad, and the average life expectancy of somebody with cystic fibrosis is only about 44 years. Ciliary motion is built on a remarkably elegant piece of intracellular architecture, the axoneme, a supportive core made of microtubules arranged in a stereotyped pattern. In most motile cilia, the axoneme has the classic 9+2 organization, 2 microtubules in the center surrounded by a ring of 9 paired microtubules. If you imagine a cross-section, it looks almost like a Ferris wheel, a central hub with evenly spaced outer spokes. If you imagine a cross-section, it looks almost like a bicycle wheel, a central hub with evenly spaced outer spokes. At the base of the cilium, this structure is anchored firmly to the cell. The central pair ends at the cell surface, but the peripheral microtubule pairs extend a short distance into the cytoplasm to form part of what's called the basal body. The basal body acts as the cilium's stabilizing dock and organizing center. The movement itself comes from dynein, an ATP-dependent motor protein attached to the peripheral microtubules. Within each outer pair, one microtubule bears dynein arms that can reach toward a neighboring pair. When dynein is activated on one side of the axoneme, it binds to an adjacent microtubule and it kind of walks along it using energy from ATP hydrolysis. That walking generates a pulling force that, in isolation, would cause microtubules to slide past one another. But because the microtubules are mechanically linked and constrained within the axoneme and anchored at the base, that sliding is converted into bending of the entire cilium. For an effective stroke, dynein activity is coordinated so that motors on one side are active while motors on the opposite side are temporarily suppressed. Otherwise, they would counteract the bend. Then the pattern reverses. The first set of dyneins quiets down, the opposite side activates, and the cilium bends back the other way. This alternating activation produces the rhythmic back and forth motion that underlies the power stroke and the recovery stroke of motile cilia. Microtubules in cilia serve another crucial role beyond mechanics. They function like intracellular transport rails. Motor proteins move cargo along these tracks to deliver structural components and signaling molecules needed for ciliary growth, repair, and maintenance, shuttling materials up and down the length of the cilium. Now, not all cilia are built for motion. Primary cilia are generally non-motile and lack the elements required for bending. They retain the 9 peripheral microtubule pairs but do not have the central pair, and they lack dynein arms. This produces a 9+0 configuration. Even though they don't beat, primary cilia are often important as sensory and signaling structures on many cell types. Okay, let's talk about flagella. Now, in humans, the major functional example of a flagellum is the sperm tail. It's the only place we see it in human cells. Structurally, it shares the axonemal core and the same dynein-driven force generation seen in cilia, but it is much longer and is reinforced by a surrounding framework, often described as a sheath of coarse fibers that stiffens the tail and enhances propulsion. Functionally, a flagellum doesn't rely on distinct power and recovery strokes the way cilia do. Instead, dynein activity propagates bends along the length of the tail, creating a more continuous, undulating wave, often described as snake-like or corkscrew-like. And that is what pushes the sperm forward. And finally, pseudopods represent a very different kind of surface tension. Extension. Rather than being microtubule-based, pseudopods are temporary, shape-shifting outgrowths filled with cytoplasm. They can range from delicate, thread-like processes to broader, finger-like protrusions. These extensions are highly dynamic. New pseudopods can form as the membrane bulges outward and cytoplasm streams into the projection, while existing pseudopods can retract when the supporting internal protein filaments are dismantled, like taking down a scaffold. And this allows the cell surface to pull back in. This continual remodeling is central to behaviors like cell crawling, environmental sampling, and the engulfment of foreign particles by phagocytes. Okay, so that's the plasma membrane. Just a basic overview of our cell boundary, the plasma membrane. It applies in so many different ways throughout our bodies, and what we're going to see throughout the course of this podcast is how much the function of that plasma membrane plays into the function of organs and tissues and cells, etc., etc. It's going to be really, really important. I'm going to end this episode here at plasma membrane. I'm going to make the next episode all about membrane transport, how we get things in and out across the plasma membrane. I hope this episode was informative for you. I'm really hoping that it helps you get your goal of a B or better in A&P. Good luck, and I'll talk to you next time. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media and is hosted by the Believe Network. Please take the time to rate the podcast and leave a review. You can also check out the content I post on Instagram and YouTube as @StudentHelp4AP. That's StudentHelp, the number 4, AP. Special thanks to my family, Bucks County Community College, and McGraw-Hill Education, where your instructor can find Anatomy and Physiology Digital Suite, my low-cost, tutor video-based digital learning solution for anatomy and physiology currently being used at several colleges and universities. If your instructor isn't using Digital Suite, let them know about it. The link is in the show notes.
If you like the show, please take a moment to rate, review, and subscribe. It really does help the show to grow. Thank you for listening.